United States Department of Agriculture.
Okay, so thank you very much for the introduction and invitation. So today I'll talk about the 3D bioprinted human tissue models, and particularly focusing on the breast cancer tissues that will hopefully reduce the need for animal models in one day to completely eliminate that in the future. So I am at Penn State, and then we focus on 3D bioprinting of living tissues and organs. So we use 3D printing technologies and create tissues, and one particular application area is in the cancer research. So before starting my talk, I just want to disclose all the agencies that I've been associated with, as well as the funding agencies that have been supporting our work so far. So before getting into cancer tissue bioprinting, so I just want to introduce what bioprinting is. You can see in the video, so you can turn off my camera if you want, so that the videos can be, seen in real time in better quality. So I can stop my video. Yeah, so in this animation, you see the cells are being 3D printed using an inkjet printer. It's the more advanced version of the paper printers that you guys have in your offices. And then instead of ink, in the cartridge that you guys use for paper printing, we use cells and deposit the living cells loaded in three-dimensional hydrogels, and then precisely stacking these printed layers, and then eventually creating an organ. And these organs, of course, with one day implanted humans. So there are some clinical trials going on with relatively less complicated organs, but for solid organs like heart, liver, kidney, we still need some time. But before getting into more clinical trials, there is one alternative application that we can use 3D bioprinting is to build 3D models. And one of them is, of course, the cancer research is the topic of today's presentation. So there are different bioprinting approaches that we use. Some of them are here. I can say we use many different bioprinting techniques in the lab. We develop our own technologies. We build our own machineries and so on. So on the left-hand side, you see a standard extrusion process. So you extrude a material with the cells laden in that material and build three-dimensional structures. So on the right-hand side, you see the inkjet printing, like the droplet-based bioprinting process, which shoots the droplets on a surface in a high throughput manner. Of course, what you see on the right-hand side is 8 times faster than the real time, but still, it's a high throughput process. And you can make things in a consistent and a more automated way, and everything is all standardized. So, of course, one application area of our research is cancer, and particularly focusing on breast cancer, as you can see here on this image on the cover of Cancer Research Journal. So the cancer microenvironment is really complex. So it's made of a number of different cells, the cancer cells, cancer stem cells, cancer-associated fibroblasts, blood vessel cells like endothelial cells or immune cells in a really complex vascularized environment. The goal of, in general, bioprinting research is to recapitulate this complexity by precisely printing living cells. Of course, bioprinting has several limitations, but we try our best to mimic this complexity as much as we can. So eventually, we want to bioprint these tissues that can be used for immunotherapy or drug testing applications. And particularly, we're focusing on the immunotherapy, testing the efficacy of CAR T therapy on these 3D printed solid tumors. So in this presentation, I'm going to show you some human breast cancer tumor models in increasing complexity. So you'll see from very basic ones, still in 3D, very basic one, to more complicated 3D models. And hopefully expectations are to reduce the need for animal models. And eventually one day, these 3D bioprinted primary human tumor models will be the gold standard for testing various different therapeutics. So in the first part of my talks, I'm going to show some published data with Dr. Unutmaz from Jackson Lab, who is an immunologist, and then he engineers T cells using CRISPR technology, and particularly It works on the mucosal-associated invariant T cells. We call them as mate cells. And then these cells are activated by a specific bacterial metabolites that are presented by a molecule called MR1. So here in this ACM, scanning electron microscopy image, you see these engineered T cells that are targeting at a single breast cancer cell. So this is an MD231 cancer cell line. And then in the first part of my talk, I'm going to show our data with the MDA cells. So this is a cancer cell line, and then I move towards the patient-derived cells as well. So here we have the very basic 3D model. It's just like a spheroid model where we make the spheroids of MDA cells, and we can also mix them with other cell types like fibroblasts, and then when we add the T cells that were engineered, as well as activate them with the supernatant of bacteria. So we see that in 2 days, the tumor, it disappears versus other groups, the tumor, they're growing. We did some cytokine and chemokine expression, and the good thing here is we found that we have seen a dose-dependent response of these cytokine expressions. For example, when you see here with the granzymes, right, as we increase the T cell density, we see some dose-dependent response. So in the second model, I'm going to run through a number of different models so that you can see what kind of 3D models have been used so far. In the second model, we put the cancer cells. These are just single cells, not in the spheroid form. And they're in three-dimensional gel called fibrin. And then here, the T cells are added on top in a device. As you can see here in the video, these T cells, they are migrating into the gel and eventually interacting with the cancer cells. So in this video, you see different zones of the device, and these show the cancer cells, and then different colors are coded for their depth. So the topmost zone, the middle zone of the device, and the bottom zone, since there is more T cell invasion into the top zone, we see less cancer cells versus the bottom one has the highest number of cells. And it's clear that we get this zone-specific response of the T cell treatment, and we see that more cancer cell in the top zone. And we also see a dose-dependent response. So if we increase the T cell, say if we double it from 4 to 1 to 8 to 1, 8 means 8 T cells against 1 cancer cell. So if we increase it, we see that the number of cells present after the treatment, the cancer cells, they are way less compared to with the 4 to 1 ratio. Another model that we had was now the tumor spheroids here in the gel. They're just not bioprinted, randomly located. And then we have the T cells. They're also mixed in the same gel. And as you can see in the video, the CAR Ts, the mate cells, they attack the tumor, and then it basically eradicating it over time. And as you can see here with the experimental group that we have, the T cells activated, you see in day 2, pretty much nothing left. And of course, some characterization of that, it's clear that with the treatment group, we got pretty much the entire volume of the tumor was gone. And then in the last approach, I want to show something that is way more complicated. And then we use 3D bioprinting. And then in this case, we create a device. So this is like a plastic device that has a channel in it. And you see little bowls here. And then these are the tumor spheroids. As you can see here, this is a zoom in fluorescent image of what we had. So this tumor spheroid was made of cancer cells, fibroblasts, and endothelial cells. And then we have the channel seeded with endothelial cells. Eventually, we have these endothelial sprouts coming out of the tumor spheroids, and then they're merging with the sprouts from the main vessel. So the anastomos here, eventually, we have the CAR Ts. You can see in this cover picture of a journal in our area. In engineering, it's a high-impact journal. So we perfuse the CAR T cells, and then these CAR T cells, they're flowing through the channel, and then they basically infiltrate into the tumor site through this anastomos capillary network, and they interact with the tumor. So in this regard, we developed again new bioprinting techniques, and one of them was aspirationist bioprinting. So I'm not going to really show the entire video and take your time. So I'm just focusing on what we did here. So we basically have a 3D gel we print, again, with the bioprinting. Could be fibrin collagen is what we use in our material. So fibrin collagen, printing them with the cross-linker layer by layer, and precisely depositing these tumor spheroids into the desired position in 3D. So, one type of spheroid, the second type of spheroid, eventually they can sprout their capillary vascular network, and then we can do various different things with the use of this technology. So we are actually making a high throughput version of this, which is now under review. So it's to speed up the process. Because right now, the process is just like printing each spheroid one by one. In this published work, with the new work, we can show that we can make it 10 times faster. So this is the procedure that we follow. Here, first, we have this device, right? We have a still wire, and then we printed the gel, collagen fibrin, and then we printed thrombin in an alternating way. And then we printed tumor spheroids locally. So we basically put them in precise locations with respect to our still wire. And then we overlay the structure and the wire with the second top half of the gel. And after cross-linking, we give some time. Then we remove the stainless steel wire, and then it's going to create an open channel. And then we inject the endothelial cells. They line up inside the channel, and then eventually we start perfusion, and then the vascular network grows. And then later we perfuse the T cells. So this is what we have. Again, the cancer spheroids made of 3 different cell types. And then we printed them, with respect to the channel. As you can see here, they can be 100 microns, 300 microns. Again, it's on demand. So then we can make them very close. And when you print very close, then they can merge. So the tumors, they merge with the vasculature, versus when you put them like this part far away compared to the close ones, you see that they're not really merging. So we have these 3D images that shows the vasculature and the tumor. So sometimes the tumor cells are invading into the capillary, again, depending on the distance. Because these tumor cells is their cell line. They're very aggressive. They grow crazy compared to the primary tumors that we have. So to test the robustness of the system, we first tried doxorubicin. It's a clinically available medication for breast cancer. And then we look at different dosages of doxorubicin. So this is 0 micromolar to 100 micromolar. This is a tumor volume. So as you can see, if we perfuse more doxorubicin, we see more cancer cell death versus no treatment. This is the tumor volume. So there is a dose responsive, tumor volume. And then in the meantime, we look at the accumulation of the drug in the tumor side. So we culture the devices for 1 day or 3 days. And then at the end of 3 days, we see a substantial accumulation of the drug in the core of the tumor. So we look at both the dose-dependent as well as time-dependent response. So we also look at the expression of some proteins. And then the interesting thing here is the samples treated with doxorubicin, we have seen, of course, the expression of less cancer-related markers as well as angiogenic markers versus the non-treated ones. The ones that were treated with very low concentration of doxorubicin had a higher expression of these proteins. So then we perfuse the CAR-Ts. So these are anti-HER2, so targeted at the HER2 receptor. So it's the epidermal growth factor 2 receptor. And then we flow the CAR-Ts through the vasculature, and then they basically invade into these capillaries, and then they infiltrate into the tumor side. So, for example, we have seen these tumors within the blood vessels as well. I don't want to say blood vessels, let's make it vasculature, vascular network, because these are not really 100% same as what we have in our native blood vessels, because our native blood vessels has multiple layers. But this is just the endothelium layer. Then we look at, again, different dosages of CAR-Ts, like 1.5 million, 3 million. And then we look at different time points, again, 1 day versus 3 days. And we had the control group, the anti-CD19. So this is for B lymphocytes, not really targeting at the breast cancer cells, but just uses a control group. And then this is our experimental group. And then as you can see here, when we treat with 3 million T cells versus 1.5 million, we see a huge difference between the right and the left columns. And the accumulation of CAR T cells, they're shown in magenta color here, is more obvious here versus the 1.5 million here. And when you look at the anti-CD19, so there was still some response. It was still toxic, but not really as substantial as the anti-HER2 one. So these are some 3D volumes measured for different treatments. As you can see here, we got time and dose responsive results. So the longer that you perfuse CAR Ts, you got better effectiveness, or the increased concentration of CAR Ts reduce the volume of tumor substantially. Then in the last part of my talk, I want to show the transition from cell lines to primary tumors that we obtain from mastectomy samples. So we got these tissues, and then we minced them by following a nature protocol. So this is just to make 3D organoids, not bioprinting in this slide. So we got various different samples, but I just want to highlight the two samples that we had. With one, the ER-PR, so the estrogen, progesterone positive and HER2 positive versus HER2 negative. So HER2 positive one, the good thing is there is the receptor that you can target that and then use immunotherapy, versus HER2 negative is really challenging for the CAR T therapies. But HER2 is more aggressive than positive is more aggressive than the HER2 negative. So we got these samples and then follow this Nature protocol paper, mince them, and then degrade them enzymatically. And then we got the single cells, put them in 3D gel, which is the base membrane extract. So this is like a 3D ECM-based gel that's commercially available. So then the cells, they grow up inside this 3D gel and then make organoids that are all random size. Amount is random. They're very inconsistent depending on the protocol, depending on your tissue type, depending on the patient, depending on the operator skills. Everything all varies. So to make everything more consistent, we use bioprinting. For example, these are some 3D grown organoids within the 3D gel. Again, this is all random size. The number of organoids are all varies depending on your conditions. So we did bioprinting. What we did is we got the single cells, and then we actually printed these in the 3D matrix. Matrix is the same matrix, the BME matrix. And as you can see here, they grow organoids over time, and then they attain a size of about 170, 175 over time. And then after the organoid formation, 10 days after the process, so we had the CAR Ts on top, and the CAR Ts, they penetrate through the gel and interact with the organoid. So these are some printed tumor organoids. Sample 1 and 2, again, sample 1, HER2 positive, you see the expression of HER2 versus sample 2, it's negative. And ER and PR is positive for both. And the K67, it's more obvious in the HER2 positive sample versus the negative one. So these are the printed organoids that we had. And then we add the CAR Ts on top, as I described. And then we wait for 3 days, and we look at day 1 and day 3 again, as we did in past work. So you see the HER2, anti-HER2 CAR Ts to HER2 positive sample. You see they eradicated the tumor, and then pretty much nothing left in 3 days, versus anti-CD19 ones. It's still affecting, not really substantially, and non-treated ones is just staying there. And sample 2 is the HER2 negative sample, the anti-HER2 one. Again, it has some effect because the CAR Ts, they have some cytotoxic effect to cancer cells, but not really as effective as the HER2 positive samples. So these are the caspase activity that shows the cell death, right? The cancer cell death. Again, we capture a dose responsive, result here. for the HER2 positive samples. As we increase the cancer cell, we got huge increase in the cancer cell death versus the anti-HER2 negative samples. You see sample 2, we did not really capture those responsive case easily, but still, compared to no treatment, there is some cytotoxicity. And then we look at the proteomics again, and we have seen a huge difference between, again, this is the HER2 CAR-T, the sample 1, HER2 positive. This is the HER2 negative one, higher expression of majority of the cancer-associated proteins with the HER2 CAR-Ts apply on the HER2 positive samples. So with that, I would like to thank all the lab members for the amazing work that has been accomplished in the last 5 years, and particularly in cancer research. And then that's it, and I can take questions if any.
USDA is an equal opportunity provider, employer, and lender.